My name is Diego, and I'll just be talking about what I've been doing at Dr. Bert's lab. And hopefully by the end, I can kind of explain more of what this image here is. So I work at the Bert lab, and we study connexins. Specifically, I study connexin 43, and there are a bunch of different kinds, but essentially connexin 43 looks like this. It's a membrane-bound protein. So there's the plasma membrane, and it loops in and out like this 4 times. And the 43 simply refers to the weight of the protein as they differ in the length of the carboxyl terminus. So this one weighs 43 kilodaltons. And so specifically, what are connexins? They're proteins that, when arranged in a circular fashion, create gap junction channels between cells. As I mentioned the subunit before, let's represent it like this. And then you can see if we arrange it in a circle like this, then it forms kind of a half a channel with a pore in the center that allows the passage of like ions and metabolites and things like that. So in this image, you can see that one half channel meets with a half channel of another cell, and they form the full gap junction channel. And so that's where the passage of information occurs between cells. And specifically, I study them in the heart, connexin 43 being the most abundant connexin in the heart. And so what that does is it couples cells together electrically at the intercalated disc between cells so that it facilitates synchronous contraction of the musculature. So again, I can kind of show you what that looks like in this little animation. So let's say we have a heart cell here, and there's another there, and then the intercalated disc is in between. And so there are a bunch of these tube-like connexin gap junction channels in between them, like so. And just imagine there's like hundreds of those. It's not really to scale. But if you have like an action potential, it can easily propagate from one cell to the next because of these connexins. And then because of that, you have contractions. There's my Hollywood level animation there. So anyway, I study this in kind of an ischemic model, which means, well, basically what I'm trying to explain here is how is connexin 43 affected by a lack of oxygen. So ischemia is a lack of blood flow, but from that you have no oxygen. So how is the protein affected by that? To explain that, I have a little infographic here. So in normal conditions, you have connexin 43 localized to intercalated discs, kind of like I showed with that animation before. So in this image, here are the cells outlined in blue, and in the green is the connexins. So you can see they're nicely in the intercalated discs for the most part. But when you have ischemia, you get lateralization of this connexin. So the proteins move to the borders of the myocytes where they can't electrically conduct as well, or if at all, when they're at the intercalated disc. So you get increased risk of arrhythmias and heart failure. So what the body does kind of in response to protect the heart a little bit is, here's the structure again of the connexin. There's a specific amino acid called serine 368 that is phosphorylated at some point after ischemia begins. And that basically confers cardioprotection to the tissue. And we know that because of this phosphorylation at serine 368, the connexin is more likely to remain at the intercalated disc. From this picture, right? The connexins being at the intercalated disc is good, and the phosphorylation of S368 makes sure that that occurs. And so what I'm doing is inducing this ischemia into a mouse heart and kind of tracking how the connexins respond to the lack of oxygen over time. I'm trying to see how long it takes for S368 to be phosphorylated. So it's 5, 10, 15, or 30 minutes. And what I'm kind of finding right now is that connexin 43 is being phosphorylated within about 5 minutes of ischemia onset. So with these images that I've been getting, you can see that after 30 minutes of ischemia, you've got nice intercalated disc connexin 43. So that shows that it's phosphorylated there. And then at 0 minutes, even though I digitally enhanced this picture, like there's not as much expression, right? There's not as much green. And then at 5 minutes, you can see that there's already starting to be kind of a lot more. Thank you, guys.